Okay, let's unpack this. We are opening up the story of the man whose name is synonymous with the very blueprint of life, Gregor Mendel.
Hmm.
He was a quiet, perpetually struggling Austrian friar who, using nothing more than, pea plants and some pretty rigorous mathematics, unlocked the fundamental laws of heredity.
We're talking about the father of modern genetics. It's a title he absolutely earned, but the story of how he earned it and how long it took for the world to notice is just incredible.
It's an incredible story of delayed recognition, for sure. So between 1856 and 1863, Mendel conducts the experiments that establish the fundamental rules of heredity.
Right. And our mission today is to really get into how he did that, and maybe more importantly, why the scientific community just ignored him for 35 years.
35 years. I mean, that's a profound intellectual lag in biology. The whole field was basically waiting for his discovery.
It was. But the real hook here, the thing that still gets debated today, is the paradox baked right into his data.
Yeah, this is the really juicy part. Before anyone even had a concept of DNA, Mendel just posits the existence of these invisible factors, what we now call genes, and uses them to predict traits with, well, stunning accuracy.
And that's the tension. That's the core of it. How could a humble monk, working without any modern equipment, produce results that were so mathematically perfect that when a top-tier statistician analyzed them in the 1930s...
A giant of the field.
A giant, yes, Ronald Fisher. He looked at the data and flagged it as being, and this is the quote, implausibly and consistently too close to the expected ratios.
So, too perfect to be true.
Exactly. It's the Mendelian paradox, and it's the mystery at the core of this deep dive. We have to decide, or at least explore. Was he the luckiest scientist who ever lived, or did he, maybe feel compelled to massage the numbers a little to make sure his revolutionary theory was heard?
To really appreciate the scale of Mendel's achievement, I think we have to start with his origins. This wasn't a man born into the aristocratic scientific elite of Europe.
Not at all. He was born Johann Mendel in 1822 in Heinzendorf bei Odrau, which was then the Silesian part of the Austrian Empire.
And his background was profoundly humble, agrarian.
Oh, absolutely. He came from a German-speaking farming family, and that farm had been in their family for at least 130 years. His early life was just characterized by working the soil. He was an active gardener, and he studied beekeeping really early on.
That practical, hands-on connection to agriculture is so key, I think, to his later success. He wasn't just an abstract thinker.
But the path from that farm to a university, it was agonizingly difficult. And it really all came down to money.
He struggled constantly through his studies at the gymnasium in Troppau and later at the Philosophical Institute of the University of Olomouc. It was a constant battle.
The sources really show the financial strain was crippling. It wasn't just a challenge. It was an existential threat to his education. He was constantly plagued by illness, partly from overwork and stress, and it forced him to take time off multiple times.
When you read accounts of his early years, the anxiety is just relentless. And education was this luxury that he perpetually risked losing.
And the family sacrifice involved is just staggering. It wasn't just his own effort.
Right. His younger sister, Theresia, this part of the story always gets me. She was so committed to his education that she actually gave him her dowry so he could continue his studies.
Think about that. That's not just support. That is an active, foundational investment in his future.
Yeah.
It shows how much faith his family had in him.
Absolutely. And this level of, desperation and family support is the critical context for his next move.
Which was becoming a monk. He took the name Gregor when he entered the Augustinian St. Thomas's Abbey in Brno.
And he did this partly because the monastery offered intellectual sanctuary and, crucially, a way to get an education without the financial burden.
So it wasn't a purely spiritual calling, at least not at first. It was a profound escape from what he himself described as the perpetual anxiety about a means of livelihood.
The monastery provided stability. It gave him the intellectual freedom that a struggling farmer's son otherwise could never have afforded. That detail just makes him so much more relatable.
It does. And you have to understand, the Augustinian order in Brno was far from being some quiet, purely spiritual retreat. It was known as a center of intellectual life and scientific inquiry.
Really?
Oh, yeah. They valued teaching, research, and engagement with the natural sciences. The abbot at the time, Cyril Knapp, was a huge proponent of scientific investigation. It was the perfect place for a mathematically inclined, nature-loving individual like Mendel to land.
So he gets there, but he doesn't just walk into a lab and start planting peas.
No, not at all. He was assigned as a substitute high school teacher. And interestingly, his primary focus after returning from his studies at the University of Vienna was physics.
And he studied under the famous Christian Doppler.
Yes, the Doppler of the Doppler effect. This is a crucial point that so often gets missed in the biology textbooks. Mendel's expertise was primarily quantitative and observational. It was rooted in physics and meteorology, not pure botany.
So he viewed the natural world as a system of measurable, quantifiable laws.
Exactly. And that explains why his approach to the pea plants was so mathematical, so different from the descriptive, qualitative botany that was common at the time.
But here's where the personal struggle continues. And I find this detail strangely comforting for anyone who's ever felt like an imposter.
The exam.
The exam. Despite the genius he would later display, Mendel failed the oral portion of the certified high school teacher exam.
Not once.
Not once, but twice, in 1850 and again in 1856.
Can you imagine? You're an innovator so far ahead of your time that you literally fail the standardized test for the job you are about to redefine.
It just speaks volumes about the disconnect between formal assessment and true intellectual breakthrough. The system couldn't measure what he was capable of doing.
But he had mentors, people who saw past those exam results. We know his physics teacher, Friedrich Franz, was key in recommending him to the Abbey in the first place.
And then there was Johann Karl Nessler.
Right, Nessler headed natural history at Olmütz and had already been conducting his own research on hereditary traits in sheep and plants. This wasn't happening in a complete vacuum.
So there was an existing, if kind of primitive, tradition of studying heredity within the Abbey's intellectual sphere.
Definitely. And people like Alexander Zawadsky also encouraged his research in Brno, possibly helping him refine his experimental design. He was building on something.
So he has the mathematical rigor from physics, the agricultural background from his childhood, and the support of the Abbey's intellectual environment. That all sets the stage perfectly for his 7 years in the garden.
But that scientific career, as we know, was ultimately temporary. It had an expiration date.
Unfortunately so. In 1867, he succeeded the long-serving abbot Cyril František Knapp, and after officially becoming abbot in 1868, his scientific work largely just ground to a halt.
The administrative duties completely swallowed his time. It's a classic story.
He traded his trowel and his observation notes for accounting ledgers and legal briefs. He became immediately and heavily burdened with these duties, especially one serious, all-consuming dispute with the civil government.
The tax dispute.
The tax dispute, yeah. Over imposing special taxes on religious institutions.
And that fight consumed the last decade and a half of his life. He felt it was his duty to protect the Abbey's intellectual and financial independence from what he saw as excessive state interference.
It was a noble cause, for sure, but it meant the world lost access to one of its greatest scientific minds prematurely. He died in 1884 from chronic nephritis, a life cut short by stress and, maybe the sheer weight of his responsibilities as abbot. Okay, let's turn now to the work that defines him. Those 7 years between 1856 and 1863. This period, spent in the monastery's substantial 2-hectare garden.
That's nearly 5 acres. It's a serious plot of land.
This period yielded the very foundation of modern biology.
And Mendel's success hinged on 2 things: his choice of organism and his methodology. He chose the garden pea, Pisum sativum.
Why peas? What was so special about them?
Well, the pea plant is scientifically ideal for a few reasons.
Yeah.
First, it naturally self-pollinates, which means you can easily get pure, true breeding lines. But you could also easily cross-pollinate them manually, which gives you complete control over the experiments.
You can play God, basically. You decide who the parents are.
Exactly. And the scale of his work, again, it just speaks to his dedication. This was painstaking work over 7 years, during which he cultivated and tested approximately 28,000 plants.
28,000? That's not a backyard garden project. This was a mass data collection effort executed with systematic, almost industrial precision.
And he was focused on specific, discrete differences. He selected 7 characteristics or traits that appeared to be inherited independently of one another.
So things like seed shape, was it round or was it wrinkled, flower color, purple or white, pod shape, plant height.
Right. And that selection was pure genius. Unlike other researchers who got bogged down trying to track complex blending traits like, say, the overall size of a leaf, Mendel chose traits that were clearly binary.
They were either or. You're either tall or short. Your seeds were either yellow or green. There was no in-between.
And that binary choice is what made the underlying mathematical patterns just leap off the data sheet. His initial focus was actually on seed shape, Was it angular or was it round?
Right. So let's walk everyone through the core mechanism, because this is where his revolutionary insight really lies. He started with what he called true breeding plants.
And that just means that if a plant had yellow seeds and you let it self-pollinate, all of its offspring would also have yellow seeds, generation after generation, a pure line.
So he takes a true breeding plant for yellow seeds and he crosses it with a true breeding plant for green seeds. This is the parental or P generation cross.
Mm.
What happens in the resulting first filial or F1 generation?
This is the first shocker. Every single plant in that F1 generation produced yellow seeds. The green trait vanished.
Completely.
Which flew in the face of the prevailing theory. Totally. If blending inheritance was correct, you should have gotten a pale yellow or a light green seed, some kind of average of the two parents. But no, you got 100% yellow.
This single observation is what led him to define the concepts of dominant and recessive.
Exactly. The yellow trait that appeared in the F1 generation, he called that dominant. And the green trait that disappeared, he called that recessive. This was the proof that traits weren't blending. They were discrete packets of information. The green information was still there somewhere, just masked.
That masking is the revolutionary concept. So he hypothesized that each plant carried two factors for each trait, one from each parent.
So when he crossed a yellow plant, let's call it big Y, with a green plant, little y,
Every F1 offspring inherited one big Y and one little y, making them all big Y, little y hybrids.
Correct. And since yellow, the big Y, is dominant, they all look yellow. The phenotype is yellow, even though their genetic makeup, their genotype, is hybrid.
But the true predictive power, the real evidence for his laws, came in the next step. He allowed those F1 hybrid plants to self-fertilize, producing the F2 generation.
And this is where the math had to be perfect. This is where the pattern reveals itself.
Because when those big Y, little y plants reproduce, they separate their factors.
That's the law of segregation. Each reproductive cell, or gamete, carries either the big Y factor or the little y factor, but never both. It's essentially a 50-50 coin flip to decide which factor gets passed on.
So if we visualize it like shuffling two separate factors, big Y and little y, from both the male and female parts of the flower and then recombining them randomly.
You get four possible outcomes. You can get big Y, you can get big Y, you can get little y, big Y, or you can get little y, little y.
And because big Y, little y, and little y, big Y are functionally the same, they're both hybrids.
And because yellow is dominant, you end up with three possible ways to be yellow. The purebred and the two hybrids.
Yeah.
And only one way to be green, the purebred recessive.
Which is how the recessive green trait reappeared in the F2 generation.
And it reappeared predictably at that famous 3-to-1 phenotypic ratio. For every one green pea, he counted roughly 3 yellow peas.
The 3-to-1 is what you see, but what you don't see, the true invisible structure he unveiled, is the underlying 1-to-2-to-1 genotypic ratio.
That's it. 1 purebred dominant, 2 hybrids, and 1 purebred recessive. That was the conceptual leap. He was quantifying the invisible.
And that's not even the whole picture, is it? He then showed the law of independent assortment.
Which is maybe even more profound. It means that the inheritance of one trait, say seed color, has absolutely no influence on the inheritance of another trait, like plant height.
So a plant sorting out whether it's going to be tall or short and whether its seeds are yellow or green, those two decisions are made completely separately.
Like shuffling two entirely different decks of cards. They don't affect each other. And this led to even more complex but still mathematically precise ratios when he looked at two traits at the same time, like the famous 9-to-3 to 3-to-1 ratio.
His approach was just unprecedented. He didn't just describe variation, he provided a mechanism and a predictive model for it.
So he presented this monumental work, Versuche über Pflanzenhybriden, Experiments on Plant Hybridization, at the Natural History Society of Brno in 1865. And it was published in their proceedings in 1866. It was effectively the rule book for how life passed information across generations.
And yet, what happened next is one of the most agonizing footnotes in the entire history of science. The publication of the rulebook led not to a revolution, but to a vast, deafening silence. We call this period, from 1865 to 1900, the 35-year sleep.
It's just crushing to think about. The paper generated, a few favorable reports in local newspapers, but that was it. That was the extent of its initial impact. It was largely ignored by the international scientific community.
Right, what times was it cited?
About 3 times over the next 3.5 decades.
That's unbelievable. So why? Why did the world look away from such a fundamental discovery?
Well, part of the problem was how it was framed or how it was perceived.
Hmm.
It was seen as a paper about hybridization, which was a common topic for botanists rather than what it actually was, a radical quantitative theory of inheritance,
It was too mathematical for the botanists.
and too biological for the physicists and mathematicians. It fell right into a disciplinary crack. We see this today as a painful example of the system failing to recognize an obscure, highly original innovator, especially one who was working outside the established academic centers like Berlin or London or Paris.
And the historical consequences of this oversight are just hard to overstate. We know for a fact that Charles Darwin, who was desperately trying to figure out how variation was inherited to support his theory of natural selection.
He was completely unaware of Mendel's work. There's a story that he had a copy of the journal with Mendel's paper in it, but the pages were uncut. He never read it.
If Darwin had held that paper in his hand, if he'd had that missing piece of the puzzle that explained particulate inheritance instead of blending, I mean, the development of genetics and evolutionary biology would have accelerated by decades.
No, absolutely. The lack of that connection is one of the great historical tragedies of science. It's a huge what if.
And despite this global cold shoulder, Mendel seemed to maintain this astonishing level of internal conviction. He even corresponded with Carl Nageli, who was one of the most respected botanists of the era.
But Nageli, who was a key scientific gatekeeper at the time, he just didn't get it. He remained skeptical. He completely failed to appreciate the importance of Mendel's discoveries.
He was stuck in the old paradigm.
He was stuck in the blending paradigm. He actually told Mendel to drop the peas and try his experiments on hawkweed instead, which was a terrible suggestion. But through all the rejection and skepticism, Mendel reportedly told his friend, Gustav von Niesel, that now famous line, "My time will come."
That's a powerful testament to his belief in the empirical truth of what he'd found. He knew he was right.
To understand why he was so revolutionary, we need to spend a moment on that dominant theory he was up against, blending inheritance. Why was that idea so intuitive to everyone?
It seems obvious, right?
It does. For many characteristics, like human height or weight or skin color, the offspring often appear to be an average of the parents. If you mix red paint and white paint, you get pink. It seems to perfectly explain what we see.
But it has a fatal flaw, especially for evolution.
A fatal flaw. Blending constantly reduces variation. If everyone blends with everyone else, eventually everyone looks the same, and that removes the raw material, the variation that natural selection needs to work on.
And Darwin knew this was a problem, so he proposed his own theory of pangenesis.
Pangenesis was was an unsuccessful attempt to solve this. Darwin hypothesized that these tiny particles called genules were shed by all parts of the body and collected in the reproductive organs, passing traits on.
It was a very complex and ultimately wrong attempt to explain inheritance without the concept of discrete, non-blending factors. It just shows how badly the scientific world needed Mendel.
And we should look at Mendel's own struggles beyond the pea plant. Because while the peas yielded these crystalline, perfect results, his experiments with hawkweed, Hericium, the plant Nageli suggested
They were a total mess.
a total mess. The results were confusing and variable. The inheritance patterns didn't follow his precise ratios at all. It was a genuine setback for him, and it likely contributed to Nageli's skepticism.
He had the right methodology, but just the wrong organism.
Exactly. The irony is so rich. The confusion was only resolved long after his death, when it was discovered that many hawkweed species are apomictic.
Meaning they produce seeds through an asexual process.
Right. They essentially clone the mother plant. So the standard rules of sexual inheritance, like segregation and independent assortment, they just do not apply. He was dealing with an organism that fundamentally defied the rules he himself had just discovered.
Wow. Had he started with hawkweed, he might never have published his theory at all.
It was a huge stroke of scientific luck that he began with Pisum sativum.
We should also just briefly touch on the full scope of his work. While we focus on the peas, most of his published papers actually related to meteorology.
That's right. He was highly systematic. He founded the Austrian Meteorological Society in 1865, and he kept meticulous records on atmospheric conditions, sunspots, all of it. It just shows his commitment to quantitative observation across all fields of natural science.
And of course, his other great love, beekeeping.
The bees. He bred them in custom-designed hives using particularly aggressive Cyprian and Carniolan varieties, which apparently annoy the other monks and monastery visitors quite a bit.
I love that detail. He called them my dearest little animals. It adds this layer of human warmth to the meticulous scientist. But the fact that he was asked to get rid of them because they were too aggressive shows that maybe his enthusiasm sometimes ran a little ahead of monastery harmony.
And we should also definitively put one persistent myth to rest.
The mouse story.
The mouse story. There's a story that Mendel only switched from breeding mice to plants because the abbot declared that observing rodent sex was unseemly for a priest.
That's just not accurate.
No. The abbot at the time, Abbot Knapp, personally oversaw sheep breeding on the monastery estate. They were not prudish about animal husbandry. Mendel's focus shifted to plants for scientific reasons, because they were a better model organism for producing clear, quantifiable results, not because of some clerical modesty.
Okay, this is the section where the story moves from historical oversight to, well, intellectual scandal. Mendel died in 1884, largely unrecognized. His work is rediscovered in 1900.
Right.
Fast forward another 3 decades to 1936. Ronald Fisher, who by this time is arguably the most important statistician and population geneticist in the world, decides to take a rigorous look at Mendel's original data.
And Fisher was in a unique position to do this. He was the person who had mathematically reconciled Mendel's discrete genetics with Darwin's continuous evolution. If anyone was equipped to understand and defend Mendel's work, it was Fisher.
So Fisher reconstructs Mendel's F2 generation experiments where that 3-to-1 ratio should appear, and he performs a rigorous statistical analysis using the methods that he himself had pioneered.
He was looking for evidence that the observed results deviated from the mathematically expected results.
Well, they should.
Right. In any real-world experiment, statistical deviation is inevitable. You have random chance, a slightly imperfect technique, environmental variables, or even just the small sample size in some of his experimental groups. All that should produce results that vary, a little bit from the perfect 3-to-1 ratio.
But Fisher found the exact opposite. He found that the ratio of dominant to recessive phenotypes was implausibly and consistently too close to the expected 3-to-1 ratio. His data showed almost no statistical variance.
It was too clean.
Way too clean. To a statistician, this is a massive red flag. It suggests that the random variation you would expect in a real-world biological system has been artificially suppressed somehow.
And Fisher did not mince words.
No, he did not. He asserted that the data of most, if not all, of the experiments have been falsified to agree closely with Mendel's expectations. Falsified. That is a staggering accusation. He used highly charged language, labeling the alleged observations as abominable, shocking, and, most famously, cooked.
He was implying either deliberate fraud or, at a minimum, an extreme, systematic, unconscious bias that rendered the data useless for true statistical rigor.
Another statistician, A.W.F. Edwards, later captured the feeling of the statistical community perfectly.
Yeah, that quote is great.
He remarked, "One can applaud the lucky gambler, but when he is lucky again tomorrow and the next day and the following day, one is entitled to become a little suspicious."
It's the pattern of perfection. Repeated across multiple different experiments, that's what raises the red flag. It wasn't a one-off lucky result.
So on the one hand, we have this powerful statistical evidence suggesting data manipulation. But on the other hand, we have everything we know about Mendel, the man, the meticulous friar, the failed examiner, the intellectually honest correspondent suggesting integrity.
And that is the Mendelian paradox. How do you resolve this contradiction between the perfect data and the good friar?
There are a few competing explanations, right?
Right. There are 3 primary explanations that attempt to resolve this intellectual knot, and each one has some pretty profound implications for how scientific discovery actually happens.
Okay, let's start with the least charitable but maybe the most common explanation. Confirmation bias.
Confirmation bias suggests that Mendel, perhaps completely subconsciously, suffered from a bias where he unintentionally favored observations that confirmed his theoretical expectation.
So he knew what the answer should be, and that colored how he counted.
Exactly. Imagine a pea pod produced 21 yellow seeds and 9 green ones. That's a 2.3-to-1 ratio, not 3-to-1. Maybe he noted a few of the paler yellow ones as green to nudge the total count closer to his expectation. Or perhaps he simply stopped counting once he reached a number that perfectly confirmed the ratio he already knew was correct.
So he's not consciously committing fraud, but his knowledge is subtly corrupting the objectivity of his counting.
That's the idea. but there's a more complex, almost ethical justification that some scholars have offered.
And what's that?
This theory suggests a conscious simplification for the sake of a breakthrough. You have to remember his context. Mendel was an obscure working-class innovator trying to communicate a radically new, counterintuitive idea, particulate inheritance, to an elite, skeptical audience that was completely committed to the idea of blending.
He had to, as one historian put it, break through the cognitive paradigms and social prejudices of his time.
And the argument goes that Mendel might have looked at his raw, messy, yet fundamentally correct data and decided that the larger imperative was ensuring his revolutionary theory was heard.
So if the raw data, with all its normal statistical noise, was too messy or complicated for a hostile audience,
It might have doomed the theory to obscurity for another generation. So maybe he made a calculated choice to simplify or polish the data to make the underlying truth of that 3-to-1 ratio absolutely undeniable and palatable to his readers.
that perspective is fascinating. It turns the supposed flaw into an act of calculated altruism for the advancement of science.
It's a very thorny ethical dilemma. It asks, can a scientist be statistically dishonest in the short term to ensure a conceptual truth prevails in the long term?
A tough question. But we have to remember that this narrative is also challenged. Not everyone buys it.
Right. That brings us to the third explanation, which is the full rebuttal to Fisher's accusation, led by modern geneticists like Daniel Hartle and Daniel Fairbanks.
And what's their argument?
They argue that Fisher, for all his genius, fundamentally misinterpreted a key aspect of Mendel's experimental design. They argue that Fisher made certain assumptions about how Mendel collected his data that might be wrong.
For instance,
They suggest that Mendel likely scored more than 10 progeny per test for certain categories, which would change the statistical expectations. A subtle change in the sample size, or the scoring methodology, which Fisher could not have confirmed because the raw data was lost, would drastically alter the statistics.
and it would essentially dissolve much of that alleged statistical imbalance.
Their conclusion is pretty definitive. They say that Fisher's allegation of deliberate falsification can finally be put to rest because the claim is unsupported by convincing evidence when you interpret Mendel's methods through the lens of a meticulous early statistician rather than a modern one.
So the data is still astonishingly good, but it might not be quite as cooked as Fisher originally claimed.
That's their argument. And the fact that this debate is still going on is a testament to Mendel's profound legacy. A man who published his findings in 1866 is still generating vigorous statistical debate among the world's leading geneticists and mathematicians a century and a half later.
So that 35-year sleep finally ended with a bang at the turn of the 20th century. Mendel's importance was realized through this simultaneous independent awakening across 3 different countries.
Yes, the 1900 reawakening was triggered by researchers who were seeking to understand the nature of discontinuous inheritance, the very mechanism Mendel had described. They needed discrete, non-blending units to explain the variation they were seeing in their own work.
And this search led to the independent duplication of Mendel's pea plant experiments by 3 scientists, all of whom published their findings within a 2-month span in the spring of 1900.
Hugo de Vries in the Netherlands, Carl Correns in Germany, and Erich von Schermach in Austria.
And the scientific integrity of the era is really evident here, because both de Vries and Correns explicitly acknowledged Mendel's priority. They gave the credit back to the humble friar whose work they had unknowingly replicated.
Yeah, while Schermach's depth of understanding of the laws is now sometimes disputed, the impact of those 3 publications was immediate and dramatic. The scientific world finally realized that Mendel had offered a genotypic understanding of heredity, that underlying factor, the big Y, little b structure, that previous studies, which had only focused on the observable phenotype, had lacked.
But this rediscovery didn't lead to universal acceptance. It immediately fueled a massive intellectual conflict that really defined early 20th century biology.
The war between the Mendelians and the biometricians.
The Mendelians, led by William Bateson, who coined the term genetics and established much of the modern terminology, they were all about these clear, discrete units of inheritance.
They were all about the 3-to-1 ratio and clear qualitative differences.
Conversely, the biometricians, led by figures like Carl Pearson and W.F.R. Weldon, focused on statistical rigor and the analysis of continuous, subtle phenotype variation like height and weight.
And they found Mendel's neat, discrete categories simplistic. They argued that life was too complex for simple 3-to-1 ratios, They thought the Mendelians were being naive.
It was a fierce academic battle. One side had a clean, simple mechanism, and the other side had superior statistical methods for analyzing entire populations.
And in a supreme historical irony, the two warring camps were ultimately reconciled through the work of R.A. Fisher.
The same statistician who later accused Mendel of cooking his data.
The very same. Starting around 1918, Fisher mathematically proved that continuous variation, like height, could be explained by the cumulative action of multiple Mendelian factors working together.
So both sides were right.
Exactly. And this successful synthesis of genetics and biometrics led directly to the modern synthesis of evolutionary biology in the 1930s and 40s, finally integrating Mendelian genetics with Darwin's natural selection theory and establishing the framework for biology that we still use today.
Now, before we get to the modern molecular age, it's worth briefly noting the tragic rejection of this science in certain political regimes.
Yes. In the mid-20th century, particularly in the Soviet Union under Stalin and later in Maoist China, Mendelian genetics was forcibly rejected. It was outlawed.
In favor of a state-mandated pseudoscience known as Lamarckism or Lysenkoism.
This ideological rejection, which claimed that acquired characteristics could be inherited and that genetics was a bourgeois science, led to the imprisonment and execution of real geneticists.
And it resulted in disastrous agricultural policies that contributed to severe famines in both nations. It's a stark reminder of the profound real-world consequences of ignoring established scientific law for political reasons.
Coming back to the science, though, the final, ultimate vindication of Mendel's invisible factors is the modern molecular proof. We know those factors are genes, stretches of DNA.
And the effort to physically identify and map these specific genes responsible for the 7 traits Mendel studied that took over a century.
It's an incredible timeline. Mendel laid down the law in the 1860s, but the scientific world didn't finish the molecular confirmation until just recently.
How recently?
In fact, it was only in 2025 that the last 3 of the 7 genes responsible for the traits Mendel observed were finally identified and mapped within the P genome.
So a complete molecular map of the 7 traits defined by a friar in his garden was only finished now. That's amazing.
It really is. And looking at the specific molecular realities just brings his abstract observations to life.
Okay, give us an example. He observed the wrinkled phenotype, the irregularly shaped seed. What causes that at a molecular level?
We now know that is caused by an insertion of a foreign piece of DNA into the PSSBE1 gene.
And what does that gene do?
PSSDE1 codes for an enzyme called starch-branching enzyme 1. The insertion inactivates this enzyme. Without it, the pea plant can't properly convert sugar into starch, causing the seeds to accumulate water, shrink when they dry, and thus wrinkle.
So Mendel was measuring the macroscopic result of a single molecular insertional mutation.
Precisely. And the yellow seed color he tracks so closely, the dominant one, that's caused by a mutation in the PSSGR gene.
That's GR.
PSSGR stands for stay green. It's a gene that typically breaks down chlorophyll to reveal the yellow pigments underneath. If that gene is mutated and fails, as happens in the dominant yellow pea, the chlorophyll degradation is inhibited and the yellow color is exposed.
What about flower color?
The white flower phenotype, a classic recessive trait, is caused by a large deletion in the PSBHLH gene. This gene is necessary for producing the purple pigment, anthocyanin. No gene, no purple, so the flowers are white.
And even plant height, which he described as either tall or dwarf.
That's caused by the PSGA3OX1 gene. This gene is crucial for synthesizing the plant growth hormone gibberellin. The dwarf plants, the recessives, have a mutation that stops the hormone production so they don't grow tall. These molecular discoveries just validate with absolute certainty that his quantitative model was mapping real, physical, biological entities.
So, after this incredible journey, what happened to the man who gave us this blueprint? Mendel died in 1884, still embroiled in those tax disputes.
And in a final tragic irony related to those disputes, the succeeding abbot, worried about potential governmental scrutiny and intervention, burned all of Mendel's personal and scientific papers.
He burned them.
He burned them. He wanted to close the book on the previous administration's affairs. We lost decades of scientific records, potentially those very raw data sheets that might have resolved the statistical paradox once and for all.
But his posthumous legacy is now immense. In 2021, for his 200th birthday, his body was exhumed.
Right. And scientists analyzed his genome, revealing that he had a predisposition to heart problems and confirming his body height at 168 centimeters.
And he's commemorated globally now. There's a Mount Mendel in New Zealand, the Mendel Polar Station in Antarctica. His time did indeed come, but only long after he was gone. What a journey we've charted today. From the financially struggling gardener whose sister sacrificed her dowry, through the 7 meticulous years in the garden, to the statistical paradox that still challenges his integrity, and finally, the ultimate molecular proof that arrived just a few years ago. Gregor Mendel's ultimate success really validates his belief: my time will come.
His work proves the power of systematic mathematical observation even when you're operating outside the scientific establishment.
The ultimate knowledge nugget here, for me, is that incredible juxtaposition of historical oversight and modern clarity. He defined his factors, the basic units of life, in the 1860s, with a theory that Darwin missed completely.
Yet it took us until the year 2025, a century and a half later, to fully identify the molecular location and mechanism of the 7 simple traits he was tracking. He saw the consequence long before we could ever see the cause.
And that brings us back to that central mystery, the Mendelian paradox. We discussed the theory that Mendel, an obscure genius, might have felt compelled to simplify his data just slightly to ensure his revolutionary and ultimately correct theory of inheritance was heard and accepted by a rigid, skeptical scientific world. It forces you to consider a really difficult choice. If your statistical evidence is technically flawed, but your underlying scientific conclusion is conceptually true and leads to a revolution in understanding, what's the right path for an innovator?
Does the quest for acceptance, the moral imperative of advancing scientific knowledge, sometimes justify actions that challenge strict scientific objectivity?
That is a deep dive for another day.